Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So, in the integration of metabolic pathways, now let's understand what is the metabolism that goes on in our body under fed condition. Let's get into the details. Now, under fed condition, that is after meals, there will be rise in glucose in the blood, rise in amino acids and rise in fats. What will happen to the glucose, amino acids and fats in the body? Glucose undergoes oxidation for energy. Glucose will undergo synthetic pathway where glucose can be converted into certain biomolecules which are essential for some of the biological functions of the cell, and also excess glucose is diverted into glycogen formation and 𝗏𝐴𝐴𝐴𝐿 synthesis. Coming to amino acids are used for a new protein synthesis to replenish any proteins that have been degraded previously. Some of the amino acids, especially the carbon skeleton, may undergo oxidation to give energy, and amino acids are used for synthesis of nitrogen containing compounds. Coming to fats are used for energy purpose by undergoing oxidation, especially in heart, because predominant energy source of heart is fatty acids, and fats can be stored especially in the adipose tissue as 𝗏𝐴𝐴𝐴𝐿, and fats are also used for formation of membrane lipids. Now this particular figure is showing you integration of metabolic pathways under fed condition. So as you see, from the intestine, carbohydrates, fats and proteins which are undergoing digestion, and after digestion they are absorbed in their simpler form, like carbohydrates are absorbed into the portal blood as glucose. Fat is digested and undergoing absorption process and reesterification, and they are loaded onto ApoB100 to make chylomicrons. Chylomicrons are appearing in the blood, and amino acids which are absorbed entering into the portal system carried to the liver and also to the systemic circulation. Now, when a person is in fed condition, insulin is increased, released from beta cells of pancreas as it is shown here, and glucagon will be decreased. Now each of these simpler forms of carbohydrates, fats and proteins, they will undergo metabolism in various organs in our body. Most of these absorbed molecules which are water soluble, first they go into the portal system and carried to the liver. From the liver they will be carried to all other organs. Now let's look at each one of them separately, and then we will come to understand this entire metabolism integration of all the metabolic pathways. This figure, I am going to explain this towards the end once I explain each individual pathways separately. Now let's look at the fate of glucose in the liver. Carbohydrate, after digestion, it is absorbed as glucose as it is shown, and increase in the blood glucose will lead to increase in insulin from beta cells of pancreas as it is shown, and increase in insulin will lead to decrease in glucagon. Now since glucose is elevated, insulin is available, so in the liver glucose undergoes glycolysis where glucokinase enzyme is induced by insulin, and all the necessary enzymes in the metabolism of glucose all the way to acetyl-CoA is induced by insulin. So glucose is converted into acetyl-CoA, and acetyl-CoA getting into TCA cycle for energy purpose. But if the person is in fed condition, retake, but if the person is in resting state, TCA cycle is saturated and acetyl-CoA making citrate is diverted into cytoplasm and goes into fatty acid synthesis and cholesterol biosynthesis. So acetyl-CoA diverted into fatty acid synthesis where insulin is going to promote all the reactions which are necessary for fatty acid synthesis in the liver, and fatty acids are esterified with glycerol 3-phosphate to make triacylglycerol, and triacylglycerol will be loaded on to ApoB100 to make VLDL, and VLDL enters into the circulation carrying the triacylglycerol, and also on the other side acetyl-CoA converted into cholesterol and cholesterol ester mediated by insulin, and cholesterol ester loaded on to VLDL. So, VLDL entering into circulation for further metabolism. Now, carbons of glucose also go into glycogen formation under fed condition mediated by insulin again. So, glycogen stores are replenished in the liver. So, this is what is the fate of glucose in the liver under fed condition. Now, some of the enzymes that are involved here in the metabolism of glucose coming all the way from intestine to the liver. So, enzymes involved in carbohydrate digestion are salivary alpha amylase, pancreatic alpha amylase, alpha glucosidase, dextrinase, maltase, isomaltase, sucrase and lactase. Whereas the enzymes that are activated under fed condition involved in metabolism of glucose in the liver going into various direction, we have glucokinase, PFK2, PFK1 enzyme, pyruvate kinase, pyruvate dehydrogenase complex, then we have glucose 6-phosphate dehydrogenase in the pentose phosphate pathway, then we have citrate lyase, malic enzyme, acetyl-CoA carboxylase in the fatty acid synthesis, HMG-CoA reductase in the cholesterol biosynthesis, and glycogen synthase in glycogen synthesis. Now, let us look at metabolism of glucose in the brain. So, the glucose that is entering into the blood after digestion and absorption in the intestine. So, glucose in the systemic circulation, it will be taken up by the brain, and brain is going to oxidize glucose all the way into acetyl-CoA, and acetyl-CoA enters into TCA cycle, completely broken down into carbon dioxide as it is shown here in the figure. That means brain is going to completely oxidize glucose for energy purpose. So, except during राजू प्रीवियस एक्सप्लेन किया था ना नेक्स्ट बोल के, उसके बाद ये एक्सप्लेन जो एक्सप्लेनेशन मैं बोला वो पूरा एक्सप्लेनेशन इधर डालना है. मैं भूल गया स्लाइड चेंज करने के लिए इधर वो एक्सप्लेनेशन डालो. सो कंटिन्यू करता हूँ वो स्लाइड एक्सप्लेनेशन के बाद वो इधर टेबल है ना, टेबल एक्सप्लेन कर रहा हूँ. ये टेबल से ये स्लाइड पहले से भी ये स्लाइड आना चाहिए. Now, except during starvation, glucose is the major source of energy for the brain. Brain and other neuronal tissue needs about 150 grams of glucose per day, remember that. And also glucose is a major precursor of neurotransmitters like glutamate, alpha, GABA, gamma aminobutyric acid, aspartate, and glycine. When blood glucose falls less than 50 mg per deciliter, hypoglycemia signs appear. These are some of the related points with fate of glucose in the brain. Now, glucose metabolism in the red blood cell. Again, after digestion and absorption, the carbohydrates releasing glucose into the circulation. So, insulin is more, glucagon is less, as it is shown. Glucose now used by the brain. Now, the glucose available in the blood is taken up by the red blood cell, and the red blood cell is going to break glucose into pyruvate into lactate, conducting anaerobic glycolysis, and lactate is taken back to the liver. Now, this is the anaerobic glycolysis that is going on in the red blood cell, and in fact, red blood cell's only energy source is glucose, whether it is a fed condition or fasting condition. So, some of the additional point here is, so red blood cell conducts anaerobic glycolysis, glucose is oxidized into lactate, and lactate enter into blood and taken up by the liver and heart, and lactate can be converted back into glucose, or lactate is simply converted into pyruvate, and pyruvate is oxidized in these tissues. And also note that glucose into 2-lactate in the red blood cell, it will yield 2 ATPs. Coming to a fate of glucose in the skeletal muscle. Again, glucose coming from the intestine, entering into the blood, taken up by the brain and red blood cell. We have seen that in the previous slide. Now under fed condition, because insulin is more, so there will be expression of GLUT4 transporters over skeletal muscle in response to binding of insulin over skeletal muscle. Now when the GLUT4s are available over the skeletal muscle under resting condition and fed state, it is going to take up glucose, and glucose is the predominant source of energy for skeletal muscle under fed condition. Now glucose undergoing glycolysis, converted to pyruvate, pyruvate into acetyl-CoA, acetyl-CoA going into TCA cycle, releasing ATPs and carbon dioxide. And when the skeletal muscle is contracting, if the person is exercising, some of the pyruvate is diverted into lactate, although it may not be a predominant process unless person is doing exercise. Some of the glucose is diverted into glycogen in the liver, thereby replenishing glycogen stores in the skeletal muscle. So some of the points here is skeletal muscle can use glucose, fatty acids and ketone bodies. Insulin allow glucose transporter number 4 over skeletal muscle to be expressed. So excess glucose is stored as glycogen, and resting skeletal muscle so primarily use glucose as the energy source, whereas exercising skeletal muscle use both glucose, fatty acids and glycogen as the source of energy. Now let's move on to see what is the glucose metabolism in the adipose tissue under fed state. Just like what I have explained with the brain, red blood cell and skeletal muscle so far, so the glucose coming from digestion and absorption of carbohydrate will be taken up by the adipocytes, and adipose tissue, it is going to allow this glucose to enter in through GLUT4 transporter, and that is facilitated by expression of GLUT4 transporter over the adipose tissue membrane because of insulin binding to insulin receptor. And glucose undergoing glycolysis releasing dihydroxyacetone phosphate in the adipose tissue, and dihydroxyacetone phosphate, it can be converted into glycerol 3 phosphate, and this glycerol 3 phosphate can be esterified with fatty acids that are coming from chylomicron metabolism, because remember, fat from the intestine will get out of the intestine in the form of chylomicron, and chylomicron undergoes metabolism. Chylomicrons undergoes metabolism in the capillary of endothelium of the adipose tissue and other peripheral tissues, and releasing fatty acids, and those fatty acids can be esterified with glycerol 3 phosphate coming from glucose, and it will be stored as white depot fat, that is triacyl glycerol, in the adipose tissue. And that's what happens to glucose under fed condition in the adipose tissue. So insulin allows glucose to get into the skeletal muscle via GLUT4 transporters. Adipose tissue uses glucose for energy if it is necessary, or excess glucose will be simply diverted into triacyl glycerol formation and stores fatty acids in the form of triacyl glycerol. That is a depot fat which is stored, which will be available for energy purpose whenever there is a requirement. Now let's look at fate of lipoproteins under fed condition. So the fat coming from the diet, that is dietary lipids, are digested and absorbed and getting out into the circulation as chylomicrons, as it is shown. Chylomicrons undergoes metabolism done by lipoprotein lipase enzyme, which is activated by insulin. Lipoprotein lipase, it is located over the endothelium of the capillary with the help of heparan sulfate, and whenever lipoprotein lipase is activated, which is usually done by apolipoprotein C2 which is present over chylomicrons and VLDLs. Let's talk about chylomicrons now. So the chylomicron catalyzed by lipoprotein lipase enzyme is going to degrade its triacylglycerol into free fatty acids and glycerol. Free fatty acids are taken up by mostly adipose tissue and heart. Adipose tissue is going to use that free fatty acid to esterified glycerol 3-phosphate to make triacylglycerol, whereas heart is going to use it for energy purpose. So the fate of free fatty acid in the adipose tissue, it is shown here as storage as triacylglycerol. What happens to the glycerol then? Glycerol is taken back to the liver, and in the liver, glycerol is converted to glycerol 3-phosphate by glycerol kinase enzyme, and it will participate in triacylglycerol formation. Note that under fed condition, even the carbons of glucose, as it is shown here in the figure, carbons of glucose converted to acetyl-CoA, acetyl-CoA converted into triacylglycerol, and that triacylglycerol is loaded on to ApoB100 protein to make VLDL. And the VLDL loaded with triacylglycerol and also cholesterol ester entering into the circulation, and in the circulation VLDL is catalyzed by lipoprotein lipase enzyme and releases triacylglycerol, which can be taken up by adipocytes and also heart muscles. So again, free fatty acids taken up by adipocytes will store them as triacylglycerol as a white depot fat, and this is what happens to the lipoproteins, especially in relation to adipose tissue, VLDL and the intestine. Now, so some of the points here: lipoprotein lipase is induced by insulin. LPL, that is lipoprotein lipase, degrades triacylglycerol present in chylomicrons and VLDL, and fatty acids from triacylglycerols are mostly stored in the adipose tissue as depot fat, and glycerol will go back to the liver for re-esterification process. Now let's look at fate of amino acids in the fed condition. So the protein that is digested and amino acids absorbed, amino acids entering into the portal blood, mostly going into the liver, some of them entering into systemic circulation. So liver, what it does with the amino acids, as it is shown here, amino acids in the liver will be used for a new protein synthesis, will be used for a nitrogen containing molecule formation, and the they can also undergo breakdown. If there is excess amino acids, carbon skeleton will be diverted into either glycogen formation or diverted into fatty acid synthesis to make triacylglycerol, whereas the amino group will go into urea formation. And the other tissues can also take up amino acids that are present in the circulation, like as it is shown, tissues taking up amino acids will use those amino acids for their own new protein synthesis or any other important nitrogen containing derivatives, and some of the amino acids may also be used for energy purpose, which is less likely because proteins are not contributing as a significant source of energy in our body compared to glucose and fatty acids. Now coming to amino acids taken up by the skeletal muscle, as it is shown here. So these amino acids in the skeletal muscle will be used to replenish proteins that were previously degraded. So this is what happens to amino acids. Basically, amino acids from the circulation taken up by various tissues to make proteins or any nitrogen containing special products in them. So here is the figure, which is a recap of all the metabolic pathways and integration under fed condition. So glucose, chylomicron and amino acids entering into the blood, they all will be undergoing the metabolic pathways as I have explained with the previous slides. So glucose used by brain, red blood cell, skeletal muscle, adipose tissue as their energy source, whereas liver is going to use this glucose and convert the glucose into glycogen and also convert glucose carbons into cholesterol and triacylglycerol, send the cholesterol ester and triacylglycerol into the blood as VLDL, VLDL undergoes metabolism in the circulation catalyzed by lipoprotein lipase and releasing fatty acids and glycerol. Fatty acids are used by the various tissues for their own metabolic needs. Now, the fat that is coming from the intestine, it is coming out as chylomicron. Chylomicron undergoes metabolism just like VLDL, catalyzed by lipoprotein lipase enzyme, releasing its free fatty acids and glycerol. Glycerol is taken up by the liver and it is used for reesterification process to make triacylglycerol, whereas fatty acids are predominantly taken up by heart and adipose tissue. Heart is going to use fatty acids for energy purpose, whereas adipose tissue use the fatty acid for triacylglycerol formation. Amino acids taken up by the tissues, skeletal muscle. So, these amino acids are used for making a new protein synthesis. So, this is what happens to the simple products coming from macronutrients that we have taken in the diet, that is carbohydrates, fat, lipids and proteins which are digested and absorbed into our circulation as glucose, fatty acids and amino acids. That's all about integration of metabolic pathways under fed condition. I hope this video has helped you in integrating or understanding the big picture of metabolic pathways. Thank you.